This recorded session is dedicated to our second module for Zoology Laboratory. We'll talk about introductory concepts on microscopy and cell structure. So these are activities 1 and 2 in the handout that I have provided or in the laboratory manual for our subject. I already uploaded the handout, and I hope that you guys will be able to read through the procedures such that we'll start right away with the laboratory proper for Saturday. We have a number of learning outcomes or objectives for this session, three from each activity. So for under microscopy, this module should help you or um, assist you, number one, in identifying the parts of the compound light microscope and the corresponding functions of these parts. Also, you must be able to focus objects, slides, specimens under different magnification using the compound microscope. And lastly, for activity one, you must be able to calculate the measurement of an object that is viewed under the microscope. Okay. Then for activity number two on animal cell structure, there are three objectives. Number one, you must be able to discuss the significance of the cell as the fundamental unit of life. So this objective goes hand in hand with our session in the lecture, our current module in the lecture on cell structure and function. Later, during your lab proper, you'll be able to observe two different types of animal cells. And by doing the lab proper, it's my expectation that you get to appreciate how, or you get to appreciate the fact that animals like us, okay, animals such as humans, our most basic unit considered to be most basic unit as living systems is the cell. Second objective is for you to describe the variations in terms of size, shape, and subcellular components of cells. And lastly, be able to list down the basic features of animal cells as applied, or as you will be able to see later on in your pre-lab, I mean in your laboratory proper. Okay, so let's discuss some preliminary concepts. So the main tool that you will be using for today's laboratory is the compound light microscope. No? And some history on this tool. The first microscope was actually created almost 500 years ago by Anton van Leeuwenhoek. The microscope he created is a microscope that can magnify objects at 300 times. No? And the intention was for him to be able to observe living organisms. No? Put it simply, a microscope is an optical instrument that is made up of a set of lenses that work together in order to achieve a certain magnification through a specific resolving power. No? Just to show you a picture of that microscope, the pre-microscope, I think I should we can say, you know, a pre-microscope that Van Leeuwenhoek invented. No? So here you can see this one. Okay, so the object I think is, or the object under observation is put here on the tip, and then the person using it is on the other side, or is peeking through at the other side. No? And some of the early drawings of Leeuwenhoek, based on his observations, look like this. Okay, figures A, B, E, and G. And if you notice in figure B, There's some indication of movement. So probably he was observing a motile cell that moved from point C to point D, and he documented that. So imagine that 500 years ago, you have this. There's the invention of the pre-microscope in a way that allowed the early scientists, the Asian scientists, to observe living systems, okay, that are very small to the very small for the naked eye to observe. Okay, and we have modern microscopes, of course. Aside from actually, our compound light microscope is not so modern anymore. We use it to observe 2D planar planar specimens, those with height and or those with width and length, no? Or just 2D, not necessarily height and width, but uh, we don't really use the compound light microscope to observe three-dimensional specimens or specimens that are prepared to observe both all three dimensions. Instead, we have more modern microscopes, the SEM and the TEM. SEM stands for Scanning Electron Microscope, while TEM stands for Transmission Electron Microscope. And these are pictures of these two types of modern microscopes. They're very different in terms of the intention of the user or the data that the user wants to get. You use the SEM if you want to observe surface structures, if you want to see how if you want to observe the topology of a specimen, you use a scanning electron microscope. On the other hand, the transmission electron microscope is used to see the super small, very, very minute object, or I mean subcellular structures. Most of the time, we use TEM to visualize subcellular structures like the organelles that are already too small to be resolved by our simple compound light microscope. Okay, there are other types of microscopes, particularly using light that has different purposes. I'll allow you to read through them, but we're not really gonna use them because we don't have them in the lab. Okay, but we can. I think for the case of our what we're using or what you're gonna use later on is what we call a bright field microscope, where light is simply transmitted through the specimen, and then it will give contrast to the specimen under observation, allowing it to be seen, allowing it to be visualized under certain magnifications. And usually using bright field microscope, the organism or the specimen that's being observed is already fixed, is already dead. No, it's not alive anymore. In bright field microscopy, we make use of stains or dyes in order to give contrast to the specimen. Um, but there are instances in which colored or pigmented cells are already visible under bright field microscope, even if you don't add a stain, even if you don't fix them. No. Then the other types, I won't really discuss more about these, but they have different purposes. No? Some of them are intended for observing the 2D rendering of your specimen while they are alive. Some of them, uh, some of these techniques you use, or some of these types of light microscopy you use to observe certain functions in living cells. No? And some of them can also be used to identify localizations of activity, or localizations of cellular activity or prominent structures within the cell. Okay. Now, in terms of function, you can simply describe microscopes as tools to help you visualize minute structures, units that are very small. size or the unit, the size ranges between less than 10 microns, no? Or 10 microns and below. So usually really, really small, really, really small specimens, like essentially cells. Okay. Other types are also used, as I already mentioned, the TEM and SEM. If you want to see more details about your specimen, you can make use of them. So we don't have TEM or SEM in our campus, but in others, like I think in Manila, in De La Salle, Manila, they have electron microscopes that can be used for such purposes. No? So let's take a look at some photomicrographs. So the one you can see here, which is colored, it's photomicrograph of a stain culture of rod-shaped cells, rod-shaped bacterial cells. Okay, so you see they are stained in order for you to observe them, in order for you to visualize them. Then we have here on the this panel is a TEM photomicrograph. And still a bacterial cell, but as you can see, this single structure here is the entire cell. Like compared to a bright field microscope, this is like hundreds of thousands in terms of the magnification. No? And then you can see specific subcellular structures that are so small, like the ribosomes, the cell membrane, the nucleoid region of the bacterial cell, DNA region. Okay. Then the panels, the photomicrographs below show us SEM photomicrographs. So here you can see, you can't really see the inside of the cell, but what you are seeing is actually the topology, the surface appearance of a particular one bacterial cell. You can see the outermost appearance. No? Okay. Now, different parts of the microscope, the bright field microscope, because we have to focus on this topic since this is the tool that you will use later on. No? So there are parts that are intended for illumination. There are parts intended for magnification, parts intended for structural support, okay? And there's other miscellaneous parts that allow you to optimize the view, optimize the magnification. I won't go I won't go about these lists or this list one by one. I won't discuss each part. I am expecting that in your biology courses during your high school, probably you've been introduced to this already. And as you've mentioned that you had microbiology in your previous years, so I'm hoping you're well aware of what parts of the microscope constitute the illuminating part, the magnification part, and the others for structural support or other functions. So you have to review them on your own. Okay. Now let's discuss some topics. Okay, in relation to the use of our bright field microscope. So first is magnification. No? Um, so we use a microscope because we want to visualize very small specimens. And inherently, cells are really small. We can see them with our own eyes, with our naked eyes only. So we make use of the capacity of bright field microscopes to magnify specimens thousands of times in order for us to visualize them, to be able to see them in larger detail or with more details. Okay, so in terms of definition, magnification refers to the process of enlarging the appearance of your minute object with the use of your optical instrument, your bright field microscope. So magnification again is enlarging the appearance, enlarging the image, okay, creating an enlarged rendering of the specimen. And in the context of our tool, the compound microscope, the photomicrograph that most likely you will be getting are two-dimensional ones. Okay. Important reminder that even though you capture specimens, they're rendering in the photo or as photo micrographs where you can see them on as bigger objects. Okay, magnifying their image does not cause their physical size to change. So always remember that. This is a very important note I want you to remember. Magnifying them from the smaller level of magnification to the highest level possible given the limitation of the bright field microscope that you have. Okay? Even though you do that, increasing magnification, you don't change the physical size of your specimen. The physical size does not change. Okay? Then another concept relative to our laboratory proper is the concept of resolution. Okay? Resolution refers to the ability of an optical magnifying instrument to reveal small details of the organism under observation. No? So simply put it, resolution is the capacity of your microscope to segregate or to tell that cellular structures are separate given a specific magnification. Okay? So, in relation to resolution, okay, there are two things I want to further discuss. So, number one is limit of resolution, and number two is resolving power. So, limit of resolution is how far apart adjacent objects must be in order for them to be distinguished as separate entities. Okay, so your compound light microscope has different objectives. You will remember that from your microbiology class. And these objectives, they have different levels or different limits of resolution. Most importantly, as you and into as a note, okay, as an important note to remember, um, the of course as you magnify you get to see more details of the specimen. So think about the fact that as you magnify from low objective, from low power, from scanner to high objective, to high power, and even the oil immersion objective, you get to see more details. So now I want you to think about the relationship or how the limit of resolution is changing. How does it change from a lower objective, from a low power objective to a high power objective? Okay. Then resolving power is the one that I've been discussing a while ago in relation to limit of resolution, where the higher, of course, you expect that the higher the magnification, the higher the resolving power. The more that you can see, the more details you'll get to see if you magnify your specimen. Now, central question, okay, review question. I want to emphasize to everyone what is the numerical relationship between limit of resolution and resolving power. Okay, so think you have as you answer the worksheet, you will encounter the formal definitions or more examples uh, illustrating these concepts. So I won't answer this question yet because we don't have it's not the post lab yet. Okay, so please think about this question. Think about the answer to this question. What's the numerical relationship between the limit of resolution and the resolving power of your microscope or your objectives? Okay? Then another concept which now relates to increasing resolving power is refractive index. So refractive index or index of refraction, it is a value calculated from the ratio of the speed of light in the vacuum to that in a second medium of greater density. So, imagine light passing through two different mediums. First is a vacuum, and the second medium is a medium with higher density relative to the initial environment. So, the refractive index is a variable that is that we can use or that we use to optimized magnification. And in terms of notation, it's symbolized by the letter N or an N apostrophe in descriptive text and mathematical equations. Okay, to illustrate that. Yeah. Okay. So you would remember that if you use the highest or the most powerful objective of a compound light microscope, you make use of an oil. Okay, you make use of an oil. And the reason behind that is because if you look at the or if you do a schematic visual, okay, to observe how light or to describe or study how light passes through from the source to the object to the optical lenses, no? You'll see that essentially, in certain instances, especially in very high power, using high power objectives, very highly magnifying objectives, okay, the complication is the working distance becomes so small or so narrow that you have to ensure that there is continuous transfer or not transfer but continuous journey or movement of light. Okay, and um, if that movement is not maintained on a straight line, or it becomes it light refracts. You remember in your physics, I'm not sure if you have physics, but one property of light is it refracts, no? And its capacity to, or its um, um, nature to refract based on the environment in which it is traveling. Okay, so um, again, I won't discuss more about that yet because we're going to answer those questions in the post lab. Okay. Then, very important in relation to, I think, the third objective for activity one is micrometry. So we get in using compound light microscope and our micrometers, we can actually measure the size of our specimens. So there are two types of micrometers. There's a stage micrometer here, number two, which is a slide. And in this slide, there on the middle of it, you can see a circle, and in within that circle, there are lines, okay, that are embedded, okay. And for our ocular micrometer, usually it's a lens, a glass looks like a button, no, a glass. What do you call this? Looks like a glass cover, but really not a cover. Okay, and similar to our stage micrometer, it has an imprinted ruler in it, an imprinted uh, 1 millimeter. Actually, no, it depends. But yeah, most likely or most of the time should be 1 millimeter. Okay, there's an imprinted ruler in both one and two that will allow you to measure the size of your specimen. No? Now, you can actually measure the size of your specimen directly using your micrometer with calibrated or with dedicated numbers or with dedicated measurements. But for our case, we will be learning how to do the calibration of our ocular micrometer. Okay? And we have a general formula for calibrating the unit sizes of our ocular micrometer. So, it's represented here in the one I am highlighting. So, the variable x refers to the divisions of the ocular micrometer that are subtended by the stage micrometer. OU are ocular units. Okay, so that's the measurement that you want to get. That's the value of each ocular unit that you intend to calculate. The idea is to be able to calculate that ocular unit value. Okay, and per objective, okay, that differs. That ocular unit size is varied across objectives. Okay? Then y, the variable y, is the divisions of the stage micrometer subtended by the ocular micrometer. Then your stage units is a standard size, which is 0.01 millimeter. Okay, so in our laboratory proper, I will demonstrate how this formula is used. But actually, part of your laboratory performance is to be able to calibrate ocular micrometers. So I won't really discuss more about that. You can do your own advanced reading. I will appreciate if you will do your advanced study on how this formula can be utilized in calibrating ocular micrometers. Okay? Sample calibration problem. I want you to think about the answer to this, and probably you can give me the answer during our lab proper. So, under the OIO, oil immersion objective, student A observed four boxes in the ocular micrometer subtended by 100 divisions of the stage micrometer. Now, I want you to, the question is for you to calculate for the ocular unit size. I'm actually not sure if I have the answer here, but yeah, I don't have. So, okay, you get to think about it. Using this formula here, okay, want you to think about the answer to this question, right? Okay, let's pause for a while. So I think this can be beyond 20 minutes already.